Hello everyone, welcome back to my channel. In this video we are going to discuss what is northern blotting. So northern blotting is a technique which we use to examine the RNA. So it is a technique which we use to examine RNA. So these are the 7 steps involved in northern blotting, starting from RNA isolation to examining. So first let's start with RNA isolation. We know that DNA and RNA, these nucleic acids are present inside the cell. Okay, if we have to get the DNA or RNA outside, first we have to lyse the cell wall and cell membrane. Talking about bacteria, we first have to lyse the cell wall and cell membrane. Okay, so once we break the cell wall and cell membrane, we will get the all the cellular components outside. And now what we need is we have to separate RNA from all other components. So we could use protease enzyme in order to digest the proteins which are present there. Like this way we could separate all other cellular fractions, and we finally will be left with nucleic acids only, DNA and RNA. In RNA isolation we use a mixture called trisol. We'll use a mixture called trisol in order to separate our RNA from DNA and protein. So trisol, it's an acidic solution. Talking about trisol, it's an acidic solution. Okay. Acidic solution, and it consists of guanidinium thiocyanate, then phenol and chloroform. We use trisol to separate RNA from DNA. In the case of RNA isolation, if you want a detailed video on RNA isolation, I'll create a separate video and upload it on YouTube. So next is the denaturation and gel electrophoresis. So once RNA is isolated, there are chances that the RNA will form a bond with itself. That is, they may get transformed into their secondary structure by forming a bond with itself. So in order to prevent that, we will denature the RNA first. Okay, we'll denature the RNA first in order to prevent the formation of secondary structures. Then what we are going to do is we will go for gel electrophoresis. Okay, here I have prepared my agarose gel. These are the places where we load the RNA, and these are called wells. Here we will load our RNA, and then we will apply a potential of 50 to 100 volts and 100 volts. So we know that DNA and RNA both are negatively charged because of the presence of a PO3 minus group in the 5' carbon. Okay, so DNA and RNA both are negatively charged. So when we apply a positive potential, it will move towards the positive electrode. We know that agarose shell has some minute pores through which the DNA and RNA move through. If the RNA molecules are smaller, it will be able to move through the pores easily. If the RNA molecules are bigger, it will be a bit difficult for it to move through these smaller pores. So the speed at which the speed and distance uh, speed at which the molecules RNA fragments move will differ. As a result, the distance moved also varies. So smaller fragments will be seen over here, and as the size increases, it will be seen to it will be more towards this side. It will be seen more towards this side. Okay. Now we have separated the RNA molecule. This is the separation step. So first we have done isolation, then denaturation, now electrophoresis. We have separated the RNA. Now we have to separated the RNA according to their size. Now we have to get the RNA outside. So we will take the gel, place it on top of a solid, and then we will put a nitrocellulose membrane here. And then what we are going to do is, using a vacuum electric field or a capillary action, we will take this RNA molecule outside, and it will be transferred to a nitrocellulose membrane. Okay. Next, what is going to happen is, once the DNA is transferred to the nitrocellulose membrane, we will add a probe RNA. Probe is nothing but a known sequence. Now the probe RNA, since it is complementary to this RNA in most of its portion, it will go and hybridize with our RNA, this RNA. This one is the probe RNA. This one is the one we have isolated. The probe will go and bind with our RNA, and after hybridization, what is going to happen is, we have labeled our probe with fluorescent tags, meaning it will emit light. Okay, we have labeled our probe DNA with fluorescent tags, that is, it will emit light. So this this RNA is now hybridized with our RNA. Now what we are going to do is, we will add washing buffer to it. That is, it is going to wash off all the unhybridized RNAs. So now we are left with only the hybridized RNA. Okay, now using auto radiography, we will be able to visualize the RNA. For example, imagine that this is our probe. If a mutation is present at this part of the of our DNA, what's going to happen here is, this will get hybridized. Okay, but in this portion there will not be any bone formation. Okay, here what is going to happen is, we will get a different signal from this part. That is, it has not been hybridized. We will get such a signal from this part. By this way, we will be able to identify the mutations which have happened in the RNA. Okay, so yes, this is what northern blotting is. So Let us see what we have discussed. First, we have isolated the RNA, then we have denatured it in order to prevent the secondary structure formation. Then we did gel electrophoresis. Then we have transferred it into a nitrocellulose membrane. Then okay, I haven't discussed this part. I haven't discussed the cross-linking part. Please pardon me. So once we transfer it, it's a very easy topic. Once we transfer it into a nitrocellulose membrane, after we transfer it into a nitrocellulose membrane, we will irradiate it with UV, or we will add some cross-linking chemicals in order to prevent this RNA molecule from diffusing. That it's a very simple thing. Once we transfer it into a nitrocellulose membrane, we will irradiate it with UV in order to prevent it from diffusing. That's it. Okay, that is cross-linking. Next one is hybridization. We will hybridize it with a probe. All the unhybridized part is washed off. Then examining. We will examine the RNA. So we are done with northern blotting. I hope you have understood everything, and thank you for watching my video. See you in the next video.